Okay, hi everyone. So we previously spoke about using the possibility of using DNA fingerprinting to identify individuals based on DNA samples, and we talked about how the general strategy um, involves involves at least these two steps. There's more to it. We're going to talk about that today, but it at least it involves these two steps typically. At least this is how it was conventionally done back in the 1980s. And the strategy is you use restriction digestion to break your DNA into a unique size profile. So individuals with the same DNA, basically the same DNA samples, gets broken into the same kinds and sizes of fragments, whereas different DNA gets broken into a different number and sizes of fragments. Then what you do is you need a way to separate out the fragments by size, and finally we talked about briefly about the visualization issue, which is that you need to actually be able to see the DNA bands as well after your gel electrophoresis. Okay. Now, we talked about how our general strategy, our ideal strategy, looks something like this. This is what we want it to look like, where we have like these clean breaks because of the restriction enzyme that causes these neat little bands, and you can kind of see here that there is a almost like a barcode-like pattern that can serve as a unique identifier for the person. So this is the crime scene sample. This is a sample from the crime scene, and you can see that the gel electrophoresis pattern in this hypothetical scenario matches well with B. So we would be able to say that that there's a match for B and there's a non-match for A here. Okay. And we also remarked how this is actually a totally fantastic scenario. What would actually happen, it looks more something like this, which is that you'll have lots and lots and lots of cuts on in any true human DNA sample. You'll have a lot of cuts for the restriction enzyme, and that would in fact lead to instead of these neat, clean, sharp signals, what you get is these smears, which are virtually useless here. And we discussed that the reason why this would happen is because we have a twofold problem here. One is to do with the sheer size of the human DNA, which is why you have so many restriction fragments forming, and the other part of the problem is how most of the human DNA is identical across people. So we can't even hope to see a difference that would be visible to the naked eye between between an electrophoresis pattern between people. So that's a problem, and we ended the video on this kind of a note that the source of the problem could be possibly that what we're doing is we're using a generic DNA dye in the scenario described above, which basically just binds to any DNA and gives out fluorescence. And we talked about how it might be possible to use a DNA dyeing method which specifically binds to certain sequences. And what sequences would we want it to bind to? Well, logically, you might think that if 99.9% of the DNA is going to be identical, what if we can specifically target the part of the DNA that actually varies across people? And could we actually get a pattern like this? This is the goal, and we kind of also mentioned that this is actually possible, something like this, not exactly this, uh, but um, but we remarked that we didn't actually get into how this actually happens. How can we zoom into the 0.1% variable region? And that is the aim of this video, to try and understand how could you use a specific DNA dyeing method to get these clean, sharp signals. Okay, so the spoiler here is that we are going to have a step 3, because the first two steps are not sufficient, and the step 3 is called is a technique called southern blotting. Okay, so make of this information what you will, and the information is that southern blotting was discovered not very far before DNA fingerprinting was discovered. So that kind of gives you an indication of how crucial this method was to this process at the time. And what southern blotting allows you to do is it allows you to selectively visualize fragments of the DNA with a specific sequence. So what this means is that you have a specific sequence in mind, and you can, instead of just adding a dye that just binds to any DNA and gives out fluorescence, you have a way to very selectively give out fluorescence when a specific sequence is found. Okay, and the result of this would look something like this, and I'm giving you a comparison between using a generic dye on top versus with sudden blotting, where we can specifically target some a fragment with a known sequence. Okay, so here you can see that that what you see is in one case is you see basically the whole of DNA after gel separated gel electrophoresis. Whereas in the case of sudden blotting, assuming that you do a similar gel electrophoresis step before this, what you get is you can only very specifically visualize the fragment which contains the target sequence. Now keep in mind, this target sequence can be anything. It can be any sequence that you know of. So as an experimenter, you might decide that you want to see you want to visualize any fragments with, say, this sequence. Okay, and there is a way to basically just very specifically add fluorescence to a DNA fragment that contains this sequence. Now how exactly this is done is beyond the scope of this video. It involves a significant amount of what I could just say is engineering magic, but it's very interesting, but it's not what we're here to talk about. So for now, just suffice it to say that you can very specifically target a fragment that contains a known sequence. Okay, so how does this help us? Well, let's go back to our dream. Let's go back to what we actually want to do. So we want to visualize only some fragment that varies in size across people, right? And we talked about how this could be part of the 0.1% of the DNA. We want to target a fragment that is presumably some part of the 0.1%, and maybe you can imagine that that a situation that we might have a situation like this, where different people where different people have different sizes of corresponding fragments. So the blue cuts here might be where the restriction enzyme cuts this DNA, and so you have in one situation you have a fragment that is larger, and when there is a match you have a similar sized fragment cut, and when there is a non-match, the fragment that you are interested in is of a different size, and that would lead to a final image like this at the end of your sudden blot. And clearly you can kind of make out that there is a match here, but this is kind of our ideal scenario. Now we have to kind of awaken to the practical constraints that we are actually working with, and the constraint here is that in order for sudden blot to work, the fragment must also contain a known sequence, right? I mean, that's what we just discussed, which is that sudden blot lets us selectively visualize a fragment with a known sequence. Now I want you to just pause and think about this for a minute, maybe, which is that these two requirements that we're talking about is they're kind of on opposite ends of the spectrum, right? So because when we talk about a known sequence, we are talking about something that is fundamentally constant. It's something that's constant across people. Okay, you need the part of the fragment is a sequence that you already know about. So for example, maybe you know that everyone has a sequence in some part of the DNA that looks something like this. So this is something that has to be constant, and let's imagine that might be somewhere here. But the same sequence also needs to be variable in size, because well, that is what makes it useful for us in DNA fingerprinting, right? So we kind of might imagine a scenario like this, where you have where as part of the fragment you have a constant part and something that varies, which leads to a difference in size. So you need both of that to happen. So maybe this is one way that this could happen. is there any other way that you could satisfy this requirement of having a sequence that is having a fragment that has a constant part, a constant sequence, and also varies in size across people? Well, it turns out yes. What if you had a situation like this? Okay, so maybe there's a part of the DNA which has these repeating units. So you take a look at the CGTA, and you can see that the CGTA is repeating. Okay, so person A has 3 repetitions of CGTA, and person B has 5 repetitions of CGTA. And if you really think about it, then this also satisfies the requirement we just discussed, which is that it has to have a constant sequence, which is common to both. Yes, it has a constant part, and it's also variable in size, right? Because if the same part repeats multiple number of times, then this is clearly going to be larger, and this is going to be smaller, as in the fragment that contains this is going to be smaller as compared to the fragment that contains this, because of these extra repetitions. Okay, and now the only question that remains is this actually seen in human DNA? I mean, is this hypothetical, very convenient situation that we've cooked up to satisfy our requirements, is this actually seen in human DNA? And it turns out yes, there is a part of the DNA that is termed as variable number tandem repeats, okay, which shows exactly this property, which is that they have a sequence that repeats over and over. What is common across people is the sequence that repeats. So the repeating units is constant across people, but the number of times the sequence repeats is variable across people, which is why you have the words variable number in the name. Okay, so now we have this almost surprisingly convenient setup that can actually work in our favor, and this is actually how the earliest versions of DNA fingerprinting, this concept is what it exploited in the human DNA to really to really selectively visualize only the very variable regions of human DNA. So as part of the 0.1% are these regions called variable number tandem repeats, and as you might imagine, if you selectively were to visualize only all CTGAs, then you might see the CTGAs light up, then that's here. But because they vary in size across people, because of the fragments varying in size across people, because of the different number of repeats, you would also see a difference in where they finally end up along in the electrophoresis gel. And sudden blotting is basically just taking it one step further and allowing you to selectively visualize only the part of the DNA that you are interested in. Okay, so yeah, that's great, and let's just kind of summarize what we've done in these 2 videos. So this is how DNA fingerprinting was done back in the 1980s. So the first step is to break up DNA into different sized fragments to get a unique size profile for each individual. Then you use gel electrophoresis to resolve the fragments by size. And finally, you selectively visualize a variable region. And typically in DNA fingerprinting, the region that you are usually interested in is some kind of repeating region for the reasons we just discussed. Okay, so it was a lot of fun making this video. So thanks for watching, and let's see